All right, so today we're going to move out of V1 into extrastriate cortex, which is everything past V1. All right. So this is higher level processing. So far, all we've talked about are detecting lines and spatial frequency. It's not really our visual perception, right? It's a pretty crude representation. We talked about the fact that our brain has to reassemble our rich visual world from the patterns of those columns firing from the different lines and angles and movements that are happening, right? So, our perception is way more complicated than this, right? And V1 is not just responding from bottom-up signals coming out of the LGN, but it can also be influenced at the V1 level by top-down signals, Right? So we call that context modulation, or one form of that top-down influence can be context modulation, which means the context that a stimulus is in modifies that column's firing pattern. It actually changes, the context can actually change the firing patterns in V1, which then influences our perception of that stimulus. All right, so let's see an example of this. Okay. And stimulus salience, how prominent a stimulus is, affects that as well. So here, we can map out a column that responds to a vertical line, okay? Doesn't respond to other angles, right? That's one of those tuning curves. And it doesn't respond to anything outside of this box. Its receptive area on the retina is only a vertical line within that box. So all of this stuff out here doesn't excite, inhibit, doesn't affect that column at all, right? And we can see, okay, when we put that vertical line there, we get a firing rate of about 33 action potentials per second, okay? Now, what happens if I now put a whole bunch of other lines out here? All of them are outside of this column's receptive field. What would you predict the firing rate of this column would be?
The same as before, since they're all outside of it.
Yeah, so if these are all outside of it, this, you would think this might be the same as this, right? Now, think about this. If, imagine that box was not there, right? Is it harder to perceive this line when it's surrounded by all these other lines than if it was just the line by itself? And so actually what happens, because there's all these other columns firing, not just the one column firing, because that's the only thing that's there, it actually influences that column and decreases it. So why is it harder to pick this out of a crowd? Well, its firing rate went down in V1. It's not as salient, it's not as prominent because of the context that it's now in. Here it's very salient, nothing else is competing with it. Here we have a bunch of other stuff competing, and that other competing factors drives down the action potentials. Now, same stimulus, look at my third example. Now, is it more prominent here than it is here? Your perception of it? Because we have these other vertical lines, does it make it stand out more than if it was just surrounded here? Perceptually it does, right? And it turns out that now, because we've added these other vertical lines, it restores that salience to it and the action potentials go up. So there's a direct, and this is really important concept that we're going to talk about over and over and over and over again in here, is that there's a direct relationship between how much brain activity is being generated by stimuli and our perception. The more brain activity, the easier it is to perceive, the more salience that stimulus has, the more prominent it is. The less activity, harder it is to perceive that. There's more interference, if you will. All right. So how does this originate? If you look at this image here, it's very hard to see a box. There's actually like a little break right here. And if we record brain activity, we get a pattern like this. In response to that, right, we initially see the image, and then there's some like perceptual stuff that happens in the next 300 milliseconds, a third of a second, and then it goes away, right? And like I said, there's really like a little break of a box here. It's really hard to see without the line there, right? But if I show you this one, where the pattern is reversed for that box, the box becomes much more salient, and now we have the same sort of initial response, but now this salience causes more activity than here. And that more activity is our perception of this different box here. It's our perception of that different box here. Right? And so all of this is happening through complex sort of connections in V1 to either suppress firing rates or to enhance firing rates and enhance our perception. Good? Yes, Steve?
I have a question about the prompting. So the picture of B that's showing more brain activity, so it'd be like just said easier to perceive like the change in the picture.
Right, because these angles are in one diagonal form, and then these are in the opposite diagonal.
So that's why it like stands out.
Yes.
Like,
Even though technically there's the same amount of white and black stimulation in the retina in both places, right? Why does this pop out and it's hard to see that box here? Because of this contextual modulation, because it generates more brain activity. Cool? Cool. All right. So we have two general pathways coming out of V1. This is getting into extrastriate cortex. Two general pathways. We have a dorsal pathway up into the parietal lobe, and that pathway is sometimes called a how pathway or aware pathway. And it's basically, I would rather refer to it as an action pathway. It's like, how am I going to deal with this stimulus? And then we have a pathway into the temporal lobe, the ventral pathway, and that's our what? What is this thing? Our identification pathway. So I like to think about this as an action pathway and an identification pathway. It helps. A comes before I in the alphabet, and so A is the top pathway, I is the bottom pathway. The reason it was called a where and what pathway to begin with was it was originally discovered in research many years ago with primates, that if you remove part of the temporal lobe, so if you give a monkey a task of discriminating between a square and a triangle, so you put a piece of food under the triangle, there's a little well under each of those, and the monkey learns, hey, the triangle is the cue of where the food is, and you randomly go back and forth putting it there. But if you remove the temporal lobe, the monkey's not able to do this. It's like it can't tell which one is the square and which one is the triangle. It can't identify them. Right? If the task is simply, hey, I have a marker, and where that marker is indicates where the food is, right? All you have to know is that the marker indicates where the food is. If we remove the temporal lobe, the monkey can still do that task. It doesn't have to identify anything other than, hey, the marker is where the food is. However, if we remove the parietal lobe, right, then the monkey can't do this task. But it can still, and we leave the temporal lobe intact, but it can do the discrimination task. So this is why it was sort of a where, because they were saying, oh, well, you can't figure out where the object is as if you don't have a parietal lobe. And the what is the identification, right? I think that think about the parietal lobe more as an action. So it's not where this is, but what's my action related to this stimulus, right? Here, my action is pick the triangle, but I can't identify which one's the triangle if you remove my temporal lobe. So I still have the ability to do the action, but I don't have the information to do the action, right? So these are separate and independent sort of pathways, right? Here, they were showing people faces of people, and in one case, the face, where the face was on the screen was indicating how they should respond to the left or right. And then in another case, they were told that the face was going to be President Biden's face and someone else's face, and you had to identify left or right based on where President Biden's face was, right? So in that, you're seeing faces. So what's stimulating the retina is still faces. So the information coming into V1 is similar, right? Well, if we look at imaging studies, when they were having to do the location mapping, like identify based on where the face is to respond, again, dorsal stream activation. When it was a discrimination between two faces, ventral into the temporal lobe. So we see this in humans too, right? Similar origination, right? The face is going to stimulate and get processed in V1, but then how that activity progresses depends on what it is we're trying to do with that visual perception. All right. So it's kind of a summary slide starting with P cells and M cells, parvocellular, magnocellular, going in here to our V1 where we're having our columns that are responding to lots of simple things, and then from there we're building up into the parietal lobe and into the temporal lobe with adding in features. And so this is another sort of more simplified pathway, right? And even though we have a magnocellular, which is mainly what kinds of perception? Movement. So those are going to be going to not simple cells, but complex and end-stop cells that have movement. And parvocellular, which is more details, which is going to be activating all of those cells, right? But there's crosstalk between there, right? Context can influence the activity there. It's not just bottom up, we have top down now. And from there we have more complex shapes in V2 and V3. We have a separate V4 color area. We have a separate V5 or MT movement area. And then we're moving into parietal and temporal pathways. All right. And this is showing it as unidirectional, but it also can flow backwards and get that top-down influences onto those things. All right. So I just talked about this, but instead of calling it a where pathway, we really should call it a how pathway. How do I do this task, or how do I respond to that? We have further evidence of this, not just from imaging studies, but when people have damage, stroke damage, for instance, to these pathways, they show what we call dissociative behaviors. They show deficits only in that pathway that's damaged and not in the other pathway. So a dissociation is when one function still works, even though another function is absent due to damage. Right? And so a single dissociation says that two functions involve different mechanisms, but they're not independent of one another. They're not independent of one another. So if one is damaged, it affects the other. So damage in V1 is a single dissociation, right? Damage to end-stop cells affects the other cells in that area, right? It affects our general perception. However, when we move out of V1 into this dorsal and ventral pathway, we get a double dissociation, meaning now once you get out of V1, if you damage the parietal pathway, you lose the ability to do those, how do I respond to this stimulus, but what is this stimulus in the temporal pathway is still intact and separate and able to function. All right? And so here's a patient that has a damaged ventral pathway. That's the identification one. So they were shown this picture and asked to copy it. And this is what they drew. Right? So they're not able to recognize this and identify it as an apple and accurately redo it. Right? But if I say, hey, don't worry about that. I want you to draw a picture of an apple. This is what they drew. Pretty good. I mean, they're not Van Gogh, but, not bad. Better than trying to copy this image. So it's not that they don't know what an apple is and they can't create a picture of an apple, parietal lobe pathway. It's if I ask you to look at something and identify it, ventral pathway, they're not able to do that. Right? Same thing here. This is a copy of this open book. If I say, hey, draw an open page book from memory, right, better at it. This, another task that we had them do is you have a target line where that arrow is, so a vertical line, and then they had a note card, an index card, and they said, okay, look at that angle of that line, and I want you to match it with the card. And this is what they did. And this is a control. So when your ventral pathway is intact, you're able to perceive the angle of that line, and you're pretty good at it. Pretty good at it, right? But then we changed, same stimulus, same task, but now instead of saying I want you to match the angle, said pretend that index card is a postcard, I want you to nail it through the slot. When they had to do an action, they were very close and able to do that almost as well as here, because their parietal lobe was intact and they were doing an action. Good? Okay, that's a double dissociation. All right, even in intact people, even in our intact brains, we can demonstrate this, right? So if you look up here, which line is longer, 1 or 2? 1, right? Okay. So down here, if you look at this example, which line is longer, on the left or on the right? The left? Okay. And so that is your estimation. So if I ask you, so if I want to measure this, if I'm not just asking which is longer, I say I want you to take your fingers and estimate how big is the line on the left, how big is the line on the right. This is what people estimate. Line on the left is bigger, line on the right is smaller, the distance between the two fingers, right? Now, and so similar to here, right? Now, if I ask you to grasp, to reach out and grasp the line, these lines are actually exactly the same. They're exactly the same length. This is an illusion with these angles, so it looks like this line only covers 2 squares and this line covers 5 squares, so it's further away, it should be bigger, right? But if I ask you to grasp, to reach out and grasp it, your fingers are exactly the same distance apart for both of them, right? Again, this illusion of context, of what's around it, affects our ventral stream, but doesn't affect our parietal stream. Because when I ask you to grasp it, you accurately do that. You don't overestimate or underestimate that line. Right? Again, it's showing that we have 2 different pathways, one of which can be affected by the illusion, and one of which can't. All right. Yes?
You said it's not an action, does that just mean like, you don't use our fingers to like compensate?
Mm-hmm. So when I ask you to reach out, You don't reach bigger for that one and smaller for this one. You reach exactly the same one. Okay, so the double dissociation means we have separate areas that are independent of each other functioning at the same time. But there's still crosstalk and modulation. So even though these look like they're far away, they're still influencing each other. When I identify what is a structure, it influences what do I do, how do I respond to that, how do I use it. So there's a lot of crosstalk back and forth between these. All right, some other areas. V4 responds to color and non-directional movement. We're going to talk about color in the second exam section. So we're going to kind of put that to the side. Middle temporal cortex responds to movement. So think about that MT and associate that with the M of movement. And it's directionally specific motion. And we can see an example here. So if we look at this, 100% of those dots are moving from right to left. There's 100% correlation. That's what's shown here. All right? If I look here, there's 50%. 50% are moving up and down, 50% are moving right to left. But we can still detect that motion, right? So the question is, how small of a correlation can we make that and we still perceive the directional movement? Here we have random, no correlation movement. There's a good example of an interactive example of this on the textbook website that's linked on the middle page, where you can go in and like change the percent of correlation and change the speed, do stuff like that, and see how it affects perception, right? So actually hidden under this is a very small correlation moving left to right, but it's very hard to detect that. If I tell you it's there, your brain kind of plays tricks and says, yeah, I can see it now. But before you knew it was there, it was very difficult to detect that, right? So monkeys with an intact middle temporal cortex are really good at this. They can train to be able to detect as little as 1 to 2% of the dots being correlated in a certain movement. If you remove the MT, those same monkeys lose some perception of that. They don't lose it all, they can still detect it, but now we have to have 10 to 20% of the dots correlated instead of 1 to 2%. Right? So the fact that there are multiple places in the visual pathway to detect motion means that if you affect just one, like the MT, you can have a deficit, but it doesn't make you blind to motion. Right? It just impairs your ability to see that. All right. The inferior temporal cortex. So middle temporal cortex, movement. Directionally specific movement. Inferior temporal cortex, the I, think about that as it. I'm identifying what it is, okay? And now we have columns that are identifying much more complex shapes. We continue that divergence out of V1 into the inferior, into V2, V3, and then the inferior temporal cortex, so that now we have, for instance here, a bunch of columns that fire to a horizontal stimulus and a bunch of columns that fire to a vertical stimulus, all converging here to create a T, right? And we can create those T's at different angles. Or it's creating a circle or a square, right? So now we're forming shapes in columns. Again, everything's still organized in columns, except now we're having more complex stimuli being represented in those columns firing. All right. So an important job that we talked about in the very first day in here is recognizing faces, and we do that by focusing in on eyes, nose, and mouth. Right? And so there are primary cells and elaborate cells. Primary cells tend to respond to a particular feature, like eyes, nose, or mouth, and the face. Elaborate cells tend to respond to complex shapes and different forms. So this cell responds best to this size circle with a line coming out here. And if we have just the line or just the circle or the circle on a smaller line, not much responsiveness. It's very specific to that elaborate stimulus. Down here, again, if the line's real thick, it doesn't like that. But if we have the line be the same length and we kind of vary the shape of the circle, it still responds pretty well to a square or a star. Yes, sir. Yeah, we're going to talk about that in a minute. Give me a sec, we'll get there.
So those bottom ones, primary cells for E, F, and G, or are those also elaborate?
Now, these are elaborate cells because they're responding best to certain combinations of features. Primary cells would be responding to like this or that, right? Or eyes or nose or mouth. A elaborate cell might respond to the pattern of eyes, nose, and mouth.
Where do they differentiate? Like, how do they tell you? How does it A to G? It's like the line could just look at the whole for original image and present is less responding, but an E to G, why is it more responding for F to G?
Because this particular elaborate cell, the most important part of it is the length and size of this line, and not as much the circular shape. It's just a big dark shape with this size line. This is a big dark shape with this size line, a big dark shape with this size line, right? All right, so some IT neurons are tuned to respond to a face. So if we look here at this image, we can see here's the spikes that are happening in that IT neuron. If we show these two images, when it's just the face, IT neuron responding. If it's just the body without the face, doesn't like that, right? So this is a cell that's responding to the presence of a face. Again, faces are very important. So if we show a monkey face, we can find a cell that responds. It also responds to a human face. As we start to eliminate components of that face, this cell is firing less. And if we make it a cartoon face, the cell goes to baseline. It's the same as if we just have black and white images, right? So it's a generalized response to faces, right? We have very few neurons that respond to a specific face. There was a research study that found an area, some cells in this area that we're going to talk about, the fusiform area, where they responded to people's grandmother's face or parents' face, and they called them grandmother neurons because they only responded to the grandmother's face and not to general faces. But that's very rare. Those tend to be really emotionally important faces to us. And in general, how do we know this face is different than my face? Is patterns of activity of neurons firing, not a single neuron coded for my face. I'm not that emotionally important to you yet. All right. So if you look up here, can you recognize these faces? Do you know who these people are? Can you write them down? 1, 2, 3, 4, 5, 6. Anyone know who this is? Raise your hand if you think you know who that is. Nope. Raise your hand if you think you know who that is. Okay, raise your hand if you think you know who that is. Don't turn your head. Raise your hand if you think you know who that is. This person? No? All right. What about now? Who's this? Denzel Washington, Jennifer Aniston, Sandra Bullock, Morley Safer, he's on 60 Minutes, John Travolta. You recognize John Travolta? And then this person, anyone recognize her? You shouldn't, because it's someone's daughter that you don't know. So it's a random picture. It's not a celebrity. It's not someone you should recognize, right? So when faces are upright, our cells fire in a way in response to their eyes, nose, and mouth that can put those faces together in a combination that's recognizable. But if I just flip the faces upside down, our brain has a really hard time doing that. A really hard time recognizing the pattern of those components. Who's in this picture? I know y'all are young, but you're not that young, right? It's former President Bush. Okay. George Bush. Okay. Is anything weird about these pictures? What about if I flip it upside down? I mean, flip it right side up. Does this look way different than this? All right, so here, the mouth is upside down. It's upside down. The eyes are upside down. Same thing here, the eyes are looking straight at you. The mouth is smiling straight at you. This doesn't feel uncomfortable to our brain because we're used to seeing eyes looking at us. And we're used to seeing mouths in this orientation, right? But when I take that same image and I flip it upside down, this is very uncomfortable to our brain. It gives us like an unpleasant emotional feeling because we're not used to seeing eyes flipped upside down and mouths flipped upside down like that. Here, right, so the mouth here is the same as the mouth here, but because the head's upside down, our brain doesn't care. It's like, yeah, well, the head's upside down, so that should look that way. All right, so in the base of the temporal lobe, there's an area called the fusiform gyrus, and this is where we find these neurons that respond to these patterns of faces. So put the F of fusiform and the F of face together, right? Associate those things together. And when we have damage to this area, which fortunately is pretty rare, because think about where this is. It's like the base of your brain, like deep. I mean, it's hard. It's not something that's going to get traumatized very easily. But there are cases of stroke, cases of brain injury that happen where this area does get damaged. And it creates a condition called prosopagnosia. Big word, it means inability to recognize faces. You can see eyes, nose, and mouth. You can detect all the features of a face, and you know that those are the features of a face, but you can't assemble them into a pattern that becomes recognizable. And what's interesting about this is that when people are affected by this, where they previously, so some people, let me just start to say, some people congenitally are born with this condition, and we don't exactly know why that happens. It's not because this area is missing or damaged, but we don't know. There's other people where this area, where they have the ability to detect faces, and then they have some trauma and this area gets damaged, and then they are not able to detect faces anymore. And what's interesting is it's purely visual. If a patient's mother walks in the room and we ask, who is that? We'll say, I have no idea who that person is. If the mother speaks, they're like, oh, that's my mom. Because the voice, the auditory voice is still associated. It's not that she didn't know her mom, she can't recognize her face. Right? It's restricted to visual. Okay. How are we doing? Good. All right. We'll finish that later. Get out of this. There we go. All right. Questions on the prosopagnosia? Okay. So, researcher, I think this is at Vanderbilt actually, wanted to know, okay, the fusiform area encodes faces, right? Could we train it? Could we train the fusiform area to recognize other things? So they created this, these objects that have some like face-like or identification ability, and they named them the greebles. And they had people come into the lab for 4 days, 7 hours a day, and practice recognizing these. Practice knowing that this is a different greeble than this is a different greeble than this, that I've seen these greebles before, and if you throw some in that I've never seen, that I can discriminate. And they got really good at it, because they spent 7 hours a day for 4 days practicing this, naming them, giving them names, and recognizing the different characteristics of all of them. Right? Okay. So then they measured responses in the fusiform area before training, and we see, okay, fusiform area is responding to people faces, not responding to the greebles. What do you think happened after 7 hours of training for 4 days? When they got really good at recognizing the greebles. Yeah, so after training, it's now responding more to the greebles. What else do you notice about the responsiveness in the fusiform area? Faces went down, right? We have limited resources. And so if you take resources away from one task and give it to another task, you might impair that other task. Right? You might impair that other task. And so it turns out that the fusiform area is not a facial recognition area, right? It's an area designed to recognize emotionally important stimuli. In this case, studying 7 hours a day for 4 days in a row made these greebles emotionally important to these people, enough so to encode them in their fusiform area, right? Other studies have looked at people who are car enthusiasts, and they can look at a car and be like, that's a 1961 Ford Mustang, blah, 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 right? They can just look at any shape of car and know what it is. Or people that are bird watchers and can identify birds very quickly and care about that. And their fusiform areas respond to cars and birds. So it's emotionally important stimuli that get put in this area. And for most of us, all of us, faces are emotionally important. But again, we have limited resources, so when we're storing other things in there, we might be limiting its ability to detect other things. All right, so we've talked a lot about the responses of specific areas, right? Specific areas. And these features may be specific or general. Specific stimuli are represented by the firing of those particular columns, those columns that are coded for particular features. But distributed coding is now when we put the activity across those columns together into a pattern. So, for instance, specificity coding for faces appears intact in people with propriocepnosia because they can describe someone's eyes and their nose and their mouth. What they can't do is put those all together in a distributed pattern that equals a particular face, a recognizable face. All right, so one of the questions is, okay, well, how do we perceive visual stimuli? Is it one or the other? What are the potential problems with this, right? And so we can kind of break this down into, we have specificity neurons that are looking at features. We have distributed processing that's looking at assembling those features into objects. All right. So here we have, one of the problems with specificity coding is if we have a neuron that fires to Bill, and a neuron that fires to Samantha, a neuron that fires for Roger, right? To code the thousands of people that you are going to remember their faces to, right, requires almost an infinite number of neurons, right, that would have to be coded for certain people. And that just doesn't seem like a good way to organize things, right? Plus, Bill, if you're looking straight on or if you're looking at the side of Bill's face, you're going to see different features, right? So how do we maintain object permanence when the actual size of the features is changing, right? So if we have a pattern of, let's just say this is eyes, nose, and mouth, a pattern for Bill, a different pattern for Samantha, a different pattern for Roger, right? Then we can take 3 neurons and code lots of different things based on the differential patterns of responsiveness, right? And so what we think is people with propriocepnosia have a problem doing this, but they can recognize the individual specific features. Okay, So we have some issues, some things that are problematic for our visual system, right? So if I'm standing up very close to you, my eyes and nose and mouth are bigger. If I'm all the way at the other end of the psychology hall, my eyes, nose, and mouth, the ratio, the size is much smaller. But you don't get me confused based on that, even though the features look different, right? Where I am in space, if I'm over here, I'm on a different part of your retina than if I'm over here. Yet, you're able to keep all that together. And then angles, right? So if you're looking at a side angle, again, your perspective is able to be maintained. So there's some more specialized neurons in the IT area, that IT area, inferior temporal area. And so we have things that are, so on the one hand, right, these are problematic. On the other hand, we need to know these, because if my face is really big, what does that mean?
Close up.
I'm close. If my face is really small, I'm far away, right? So we need to know something. We don't want to just ignore size. You want to know where I am. Am I over here or am I over here? Location matters. You want to know which angle of my head you're looking at. So we want to have some specific neurons that fire to faces of different sizes, because that's an important thing for us to detect, and where they are and what view. But we also have neurons that are, when we say invariant, they ignore that characteristic. So specific neurons give us important cues about things, but invariant neurons maintain our perception in light of differences in size, or differences in angle, or differences in location. All right, so again, all these neurons serve a function. So here we see a neuron that is I'm sorry, let me back up. So if this neuron is firing equally to this space, what type of a neuron would you classify that as? View invariant, right? Angle invariant. The angle of the head doesn't matter, right? What about this neuron? What does it respond to? The upright face, but not to it rotated in different angles. So that would be a view specific or angle specific neuron, right? And so combining these allows us to maintain perception in light of changes, but also recognize the position of that face or that object, any object really. We're just using faces as the example here. All right. If we look in people at the activity of their fusiform area, again, these are ERP recordings, so we're looking at the first 300 milliseconds of activity, right? And we're looking at the fusiform area. It has this initial, hey, there's a stimulus there. If it's a face, my activity is going to go way up. If it's anything that's not a face, my activity is not going up. To animals, houses, or objects, right? Now, so here's our objects down here. If I have similar things, human faces, cat faces, line drawings of faces, profiles of faces, all of those are strongly activating the fusiform area. They're all being recognized as faces in that distributed pattern, not objects. So there's specific responses. Okay, this is an area looking for eye, nose, mouth patterns, but it really doesn't matter what type of pattern it is, even if it's an animal face versus a human face, all of those are getting generalized in that area. Yes? The fusiform area.
So what's the area you just references?
The fusiform area. So this is faces and non-faces, right? This is types of faces, even if it's an animal face, right? Now, if it's your cat's face, you're going to recognize, oh, that's Tabby, my cat. If it's some other random cat, you're going to be like, that's a cat's face. you're still gonna fire it, it's a catch space, you're just not gonna identify what it is because it's not encoded as a memory, right? So at some level, these patterns are reaching into memory areas and associating those patterns with who or what that is. All right. So one of the questions is, how does all this develop, right? Are we born with the fusiform area destined to become this area of people? And we don't know the answer to this question, right? But presumably, yes, in most of the people, the fusiform area does this specific job. Therefore, it must be sort of set up to be encoded with information. Now, the faces you recognize and the faces I recognize are very different because we've had different life experiences, and I haven't met the people you've met, and you haven't met the people that I've met. So even though our fusiform areas are genetically coded to do this, what's coded in there depends on experience, depends on nurture, right? Obviously, recognizing faces has a strong survival mechanism, evolutionary mechanism. So you could see why an area of the brain would be devoted to that particular thing, right? So in general, like everybody's V4 does color processing. So there's some genetic code that says, hey, this area is for color processing. However, if you never perceive the color red, and it won't get encoded in that particular area. So while we have areas that are genetically programmed to do specific things in the visual path, in the visual system, if they don't experience stimuli, then those stimuli don't get encoded in those areas. All right, so some, again, evidence of this. We want to, again, one of the things that we're striving to do in here is to correlate neural activity with perception, and changes in neural activity with changes in perception. Just like we talked about with context modulation at the very start of this lecture. Right? So here we have little like bent paperclip things, right? They're slightly different. We can train a monkey to recognize this one and discriminate that this is not the same as this over time, right? And so when they first start out, they are not good at this, and they don't have increased firing. They don't have differential firing rates for these different stimuli. They don't mean anything to them. But after reinforcing this one and not reinforcing all the others, the monkey starts getting really good at recognizing this. And so after training, right, their psychophysical choice goes very high, and so again, we generate like a tuning curve where they're able to recognize this stimulus. They might get confused with very similar stimuli, but as we make the stimulus different, that drops down. And if we look at what happens in the inferior temporal cortex, we now find a column dedicated to firing to this and not firing to these other stimuli. So how did the monkey get better at that task? It got better because it trained an area of its brain to recognize that and respond to that, to allow it to have that perception. We good on that? That's real. This is like, again, the crux sort of part of this is that we can look at changes in brain area that correlate to changes in behavior and say that behavior, the ability to do that behavior is because we now recognize the firing rate in these particular parts of the brain. All right, so there's a combination of both of these, right? We have areas that are designed and reserved for different feature detection, P1. We have specialized responses, but the responsiveness of those neurons relies on our experience, and even after development, we can encode new stimuli. Your brains are fully developed pretty much right now. Right? But you meet a new person this summer and work with them, you're going to encode their face and remember their face, right? So we have plasticity, we have the ability to do these sorts of things. Okay? So again, it seems like the most efficient use of neurons is this distributed coding, to have specific areas that are going to break down things and then re-encode them. Yes, Drew.
Do we have, like, we're talking about limited resources, is there a limitation to how many faces we can recognize?
Not that I'm aware of, right? Probably, but no. And I'm not aware of research that's looked into this. All right, don't get excited because we have 10 more minutes. I want to finish that that video, which is the start of the next